Hello and welcome to our next lesson in AP Biology about nucleic acids. What you're going to see from this document is that it doesn't have any open-ended questions or anything skeletal about it. It's just an information packet. So there's not much to do other than to read over it. So give me a second and I will share it. So as you see here, this is a Word document that I'm sharing with you. So nucleic acids, we're going to be focused on for the biochem unit for unit 1 on the chemical structure and polymerization. There will be a separate unit when we get into DNA and RNA and molecular genomics, which will be in unit number 6. And that way we'll get into more about how the structure and function relate to its actual, how it actually functions. So anyway, the main types of polymers of nucleic acids are RNA, which is ribonucleic acid, and DNA. And they're both made up of monomers called of nucleotides. And a nucleotide has 3 components: a pentose sugar, that's either deoxyribose or ribose. And if you remember from our when we learned about carbohydrates, that this top oxygen of the ring is what you use as your basis to then count the carbons. So there's 1, 2, 3, 4, and then the one that's kind of the antenna is number 5. So off of carbon number 2 in deoxyribose is just a hydrogen, whereas in ribose it's a hydroxyl group. So it's got less oxygen, therefore it's deoxy. You're also going to have a phosphate group attached to carbon 5, so that would be up on the antenna right here, and then a nitrogenous base attached off of carbon number 1, so that would be off of right here. There are 2 types of nitrogenous bases, pyrimidines and purines. Pyrimidines are single ring bases and purines are double ring bases, and they are nitrogenous bases in that they have nitrogen along with the other carbons. And you can see they have carbonyl groups as well. So they are fairly polar in terms of their structure. And if you look at the full structure of a nucleotide, you can see there is the phosphate group off of carbon number 5, the nitrogenous base off of carbon number 1, and then this is a deoxyribo because you can see the hydrogen is by itself right there instead of with an OH. The formation of a polymer happens when the hydroxyl group off of carbon number 3 and the OH of the phosphate group interact and form a phosphodiester bond. So this is the phosphodiester group, so it's a carbon with the phosphate and another carbon on the other side. So it's a phosphodiester, and it's another example of dehydration synthesis. So you can see where it's in red, those are the 2 that'll interact. The hydrogen off of the hydroxyl group will leave, and then the OH off the phosphate group will leave. As I said, there's 2 different types of polymers, the DNA and RNA. So DNA is a double helix. So if you see, this is the one strand, and then there's another strand that runs in the opposite direction. And so the polymer goes along the one side between phosphates and sugars. They make what we call the backbone. And then the 2 halves of the polymers of the double helix are held together by hydrogen bonds between bases, and specifically the base T, thymine, bonds to adenine, cytosine bonds to guanine, adenine to thymine, guanine to cytosine. And you can also see that there are different numbers of hydrogen bonds. So between guanine and cytosine are 3 bonds, between thymine and adenine are 2 bonds. That's for later, that'll have more importance. This is a picture of ribosomal RNA. Ribosomal RNA is actually single-stranded. You can see it has a single 3, this 3' and 5' refer to the ends. So this is a 3', 5', and then another 3' and 5'. So you can see this is actually just, in the case of ribosome, it's just folded over many, many times, but it's a single strand. And then this is transfer RNA, again, a single strand, you can see if you follow it along, and then it's actually just folded up, and there's some hydrogen bonding in between. So somewhat similar to proteins, and in many cases, like the case of ribosomal RNA, it actually has some of the same sorts of functions that a protein could have. It has an enzyme function to it. Then we have nucleotides and oligonucleotides. You have adenine or adenosine triphosphate, or ATP, and you should probably know that as being one of the key things in your body that is going to provide energy, and it does that by usually through functional group transfer of this last phosphate group right here on the end. It will add that to some target molecule and change the function of the target molecule. There's some other things like riboflavin and nicotinamide adenine dinucleotide. And these are things that are going to be found as other components in metabolism, so vitamins or other things that will add to how things work in your body. But obviously we know about ATP, and we know about RNA and DNA in terms of their function in protein synthesis and the maintenance of the genetic code. So that's usually what we're going to be focused mostly on, but we certainly will be talking about ATP and NAD and riboflavin when we get to photosynthesis and respiration. So that's it for the nucleic acids during the Unit 1. Again, we will revisit this later in Unit 6. So for now, just focus on the components of the monomers and how they are put together, and how it polymerizes, and a basic idea of how it might function in the case of DNA just functioning as genetic material, and RNA supporting in protein synthesis. All right, have a good rest of your day.